This is a production of Cornell University.
Thanks.
No problem.
Yeah, thank you for the introduction, Savannah. I'm very excited to share my research with you today on the topic of harnessing tomato diversity for structural variant and gene expression correlation analyses. So tomato originated in South America, where wild relatives can still be found today. And two of the orange-colored tomato species, or wild species, Solanum cheesemaniae and Solanum galapagense, were originally the only two tomato species found in the Galapagos Islands. The red-colored Tomato pimpinellifolium is the only wild species that is red-colored, is native to Peru and Ecuador, and was domesticated into the Solanum lycopersicum variety Cerasiforme, which was then further improved into the Solanum lycopersicum variety Lycopersicum, which is now grown in more than 140 countries around the world, with a production of more than 180 million tons of tomato in 2018, which makes tomato the second most important vegetable crop after its closely related potato. Tomato shows a very wide morphological diversity. As you can see in the left picture, fruits can come in all kinds of forms and colors, and also the plant itself has quite wide morphological diversity, as for example you can see in the leaf pictures on the right. Besides morphological analyses, a vast number of tomato accessions from different species have been genotyped, providing large amounts of information on tomato genomics. In the left here, you can see the circle, which is a phylogenetic tree made based on genomic information from several hundred of tomato accessions from different tomato species. And high-throughput phenotyping, or the increased use of high-throughput phenotyping, accelerated the identification of natural genetic variation between tomato accessions, mostly showing a lot of short single nucleotide polymorphisms, or SNPs, which can be used in GWAS, for example, to demystify the genotype to phenotype equation. So the predominantly used short read sequencing skews the identification of natural genetic variation towards single nucleotide variants or short indels. However, most of the diversity observed in tomato is ascribed to the selection of rare alleles with a large effect size. The majority of genomic differences are attributed to structural variants, which in the genotype to phenotype equation have been disregarded. So let's take a step back and look at what structural variants are. So these are large deletions, insertions, duplications, or other kinds of structural chromosomal rearrangements, with large meaning like 50 base pairs or larger usually. So structural variants can cause changes in the composition or position of cis regulatory regions, or can directly alter the gene copy number, all of which can lead to quantitative changes in gene expression, or even to changes in phenotypes. And what's interesting as well is that when you look at SNPs, you oftentimes do not identify structural variants, or like the SNPs that are identified are not within structural variant regions. So 3 recent papers looked at structural variants in tomato. The papers are listed in the bottom. And the figures here are results from the Alonghi et al. 2020 paper. They looked at 100 tomato accessions from different species, which are shown on the x-axis of the largest image here. And when we look at the count, we can see that the number of structural variants in a single accession range from around 2,000 to more than 45,000 structural variants. And most of the structural variants occur in tomato wild species, here with the pink and red dots, then fewer in the Cerasiforme in green, and even fewer in the Lycopersicum in these purple and blue color dots, which is consistent with the genetic loss from the wild species to the domesticated Cerasiforme to the improved Lycopersicum. Structural variant types mostly are in pink deletions or in blue insertions, but also inversions, duplications, and translocations have been found. When looking at the detection frequency, most structural variants are rather low in their frequency. However, when looking at the 35,000 annotated genes within tomato, 95% of the genes have at least one structural variant associated to the gene. The size or length for the structural variants is rather short, like 30 to 50 base pairs, although usually structural variants are only counted starting with 50, so it would be like 50 to 500, 500 to 5,000, but there also are structural variants that are even larger than 5,000 base pairs. Around 50% of structural variants are in intergenic regions, which is here in pink, and the other 50% are associated with genes. So the other, like the different colored parts of this bar plot, and those can be separated again into structure variants within genes or within the 5 kb region upstream or downstream of genes. The studies also identified or found that structural variants do impact gene expression, and when looking at specific structural variants, they also found that it affects the phenotype. So we know, especially from the 3 studies, that there's extensive amount of structural variant sequence data, but the amount of gene expression data is limited, with most of the gene expression data that was used in those 3 studies actually comes from a study from 2018. So that's quite limited. However, the identification and the understanding of the effect of structural variants on gene expression is valuable for resolving their functional impacts on gene expression as well as phenotypes, and to understand how structural variants play a role in this genotype to phenotype equation. So the goal of my research is to generate a large gene expression data set to correlate the expression profiles with structural variants in tomato. My workflow includes 4 major steps, with the first 2 steps being material collection, the material processing, and RNA sequencing. Those are the 2 steps that I'm mostly working on at the moment, although I'm still kind of in the material collection phase. Once I get the RNA expression data, I can correlate this with the structural variants, which I know based on the 3 studies I mentioned earlier. And the correlation analysis will be based on a pipeline that was described in the Alongi 2020 paper. Finally, I can do, or I want to verify the associations between gene expression, structural variants, and also phenotypes via sequence editing, whereby I want to focus on fruit ripening genes. So the selection of my genotypes and tissues for my gene expression dataset includes includes 178 accessions from the listed species here, with 8 and 9 accessions of the Gismaniae and Galapagensis species, 34 of Solanum pimpinellifolium, 62 Cerasiformi, and 65 of the modern Lycopersicum, which can be further separated into fresh tomato, processing tomato, and also heirloom tomatoes. The tissues I'm sampling are leaf material, flower material, and then fruit material from 3 different developmental stages, including the mature green stage, breaker stage, and breaker +7. And for the fruits, I'm only sampling the pericarp tissue, which in the diagram here you can see is the green colored tissue. I also have 3 biological replicates for each of these samples, which calculating everything together gives me 2,700 samples, which I think is a great and a huge number, but also a great amount for getting a good data set together. Okay, so the material processing includes RNA extraction and library preparation based on poly-A enrichment. The RNA sequencing will be done on an Illumina HiSeq X10 with paired end sequencing, 150 base pairs read length, and 10 million reads per library. Besides the RNA or gene expression profiling, I'm also going to look at carotenoids for carotenoid profiling, and I'm going to send the samples to Fernie at the Max Planck Institute for volatile analyses, and I also send leaf material to the Sinha lab at UC Davis for leaf morphology analyses. So I don't have any results yet, but I'm happy to share these with you next year, so tune in then. I would like to thank the Giovannoni lab, especially Jim, for providing the research and the funding for the research, and Eamon for helping a lot with the plants and the material processing. With that, I would like to take questions.
Thank you, Anna, for that great talk. Wonderful. If anyone would like to ask questions, please, of course, feel free to put them in the chat or unmute yourself and ask. I have a question, Anna. Do you know, you mentioned that SNPs that people are using are usually not found within these structural variants, right?
Mm-hmm.
Do you know why that would be?
To be honest, no, I don't. Okay. I guess that maybe if there's like the larger structural variant, maybe there's several bases next to another that are changed so that it's not actually a single nucleotide polymorphism. It might be also that there's like regions of nucleotides that are changed, with like one being changed and then a few next to it are the same as the reference, let's say. But I guess they're less frequently identified because you have several bases next to another that are different.
Yes. Ed Buckler asks, indels are generally younger. It's more of a comment than a question. He says, indels are generally younger and have a shorter coalescent.
Okay. Thanks, Ed.
And Jean-Luc has his hand raised, so please feel free to ask your question.
Thanks. So if I understood, the materials that you're using are all ones that were studied in that Alongi study, and you're gonna.
So the accessions, the genotypes that I'm looking at have all been sequenced. So we have the like the sequence data, the genome sequence, with quite high like good depth. So they identified the structural variants, but the gene expression is new. So they didn't. The research from Alongi is based on like the 2018. They only looked at orange fruit samples.
So I mean, but did you choose any of them on the basis of phenotype, or is this a subset of the Alongi, or this is everything?
So the phenotype or the tissues that I'm sampling are based on our interests on fruit ripening genes in general, which is why we look at specifically fruits, which is our main goal, at different developmental stages. And those 3 stages that I picked are stages that are like common to look at. So there are like a few papers where they have limited data on expression data, but limited expression data, but then usually on these stages. And the leaf and flower are mostly to complement the fruit tissues. Does that explain your question?
I was wondering. So whether the accessions that you're working with, is this the whole set of accessions from the Alongi study, or is it a subset? And if it's a subset, how did you base your subsetting on?
Okay, yeah. Sorry. So the accessions that I chose are all chosen based on genome sequence available for those. And I think not all of, so Alongi looked at 100 accessions, and I'm looking at more accessions, but not at all of his. And I think that's because we limited the accessions also to which seeds we could get easily.
Okay, great, thanks.
Yeah.
This has been a production of Cornell University, on the web at cornell.edu.